Hello students and welcome to the study portal again. The topic of today's video is cell physiology. This video is in continuation with the previous video regarding cell physiology. In the previous video was the part 1 of the cell physiology. In that video we discussed about cell, the structure of cell and the various functions performed by the cell. Alright. So in the previous video we covered the functioning like transport mechanism, the active transport we covered in that video. We were discussing the passive transport. In passive transport, in the earlier video we discussed about the simple diffusion. Now today's video will be highly emphasized on certain other functioning performed by the cell, such as the different types of passive transport mechanisms of cells and different functions such as osmosis, tonicity. We will discuss about the different concentration of this different concentration that are present in the cell. Alright, so without delaying, let's start the video. So the topic is facilitated diffusion. Facilitated diffusion is a type of passive transport. As we are aware that passive transport is the movement of any substances across biological membrane from an area of higher concentration to the lower concentration area without the external energy. Alright. So the passive transport is the movement of substances from higher concentration to the lower concentration along with the concentration gradient without the requirement of any external energy. Now what is facilitated diffusion? So the facilitated diffusion is the transport of substances across a biological membrane from an area of higher concentration to the lower concentration with the help of transport molecule. So what happens in the passive transport? Let me clear you with an example. That first of all, let's look at the definition, that it is the movement of substances from higher concentration to lower concentration with the help of a protein. So a protein will help, or transport molecule will help to move the substances from higher concentration to lower concentration. Alright. Examples of biological process that entail facilitated diffusion are glucose and amino acid transport, gas transport and ion transport. So all these type of transport occurs with the help of facilitated diffusion. It is important because it regulates what goes and what goes in and what goes out of the cell. The plasma membrane is the cellular structure that is responsible for the selective movement of substances. We all know this now. Alright. So it is through this facilitated diffusion, the biological molecules or ions across a plasma membrane move or are transported with the help of transport protein located in the plasma membrane. Alright. So let's just have a look at the characteristics of the facilitated diffusion. It is a type of cellular transport where substances move along with their concentration gradient. The difference in concentration between areas creates a gradient that incites substances to inherently move to be distributed between two areas to achieve equilibrium. Because the movement is downhill, that is from higher concentration to lower concentration, alright, chemical energy is not directly required. What drives facilitated diffusion, just like the other types of passive transport, is kinetic energy. So here in this kind of facilitated diffusion, kinetic energy is required, chemical energy is not required. Alright. So why facilitated diffusion is important, or is it has a characteristic? Because it is the need of assistance from a transport protein lodged in the plasma membrane. Alright, so here you can also see in this picture that how glucose is moving in, and glucose, then in the second example glucose is moving inside the kidney, and the sodium ions are also moving inside the kidney, and in the antiport the ATPs are going in, and ADPs are going in from the other side of the membrane. So this is uniport, symport and antiport explained here. Alright, so the cell membrane is permeable only to a few molecules that are smaller in size and non-polar. Therefore, facilitated diffusion with the help of transmembrane protein is important. As I told you, a transport molecule is required in the facilitated diffusion that will transport the substance from higher concentration to lower concentration. In the facilitated diffusion, energy is external energy such as chemical energy is not required. The facilitated diffusion is carried out with the help of kinetic energy only. Moving on, now talking about filtration, that how filtration is achieved in the cells. So filtration is the process of separating solids from liquids and gas. As you can see here, it is written that filtration is the process of separating solids from liquids and gases. So filtration is another passive process of moving material through a cell membrane. While diffusion and osmosis rely on concentration gradients, filtration uses a pressure gradient. Alright, molecules will move from an area of higher concentration, higher pressure to an area of lower pressure. It is non-specific. This means that it does not sort the molecules. They pass due to pressure gradient and their size. If molecules are small enough to pass through the membrane, they will. The force that pushes the molecule is termed as the hydrostatic pressure. Let's understand this with the help of an example. Let's take the example of making coffee. Think of the coffee filter as the cell membrane and the coffee grounds, flavor and caffeine as the molecules. The pressure is exerted by the water from the machine. It forces materials through the coffee filter into the coffee pot. Small molecules like caffeine, water and flavor pass through the filter, but the coffee grounds do not. Because they are too big. If you poke holes in the filter, the coffee grounds would end up in your coffee. The coffee filter represents the filtration membrane, which is typically a layer of cells. So this is how filtration works in your cells as well. Alright, what happens that in this picture you can see that there are holes, because so the feed is not able to pass if there won't be any holes. If there will be holes, if you poke holes in the cellular layer, then the feed or the substances that need to be transported will get transported due to the pressure gradient. Due to the pressure gradient, the substance will move from the higher pressure gradient to the lower pressure area. Alright, so on this basis, the filtration works on the basis of pressure gradient. It is one of the main methods used for the capillary exchange. Blood pressure provides the driving force or hydrostatic pressure to force material out of capillaries to cells or to form the filtrate. Hydrostatic pressure is countered by osmotic pressure. Remember, osmotic pressure is created due to increased solute concentration and will pull water toward the area of higher solutes. Alright, so this is the example where the filtration is carried out. In the example of capillary exchange or in the blood pressure mechanism, the filtration process works. So kidneys are also an example of biological filter, because kidneys help in the filtration of waste material. The plasma is filtered and the waste materials are excreted out of the body. Alright, so by the glomerulus, blood is filtered and necessary molecules are reabsorbed So they will move from the higher pressure to the lower pressure. In the process of filtration, cell membrane permits only those substances which are soluble and could easily pass through its pores. Here you can see in this picture also that the substances which are of the smaller size can only be transported, whereas the substances which are of higher size cannot move because of the pore size and their size. Alright, so filtration is the process of separating solids from liquids and gases. Now the next term is osmosis. Osmosis is also a type of passive transport. In this process, water and other molecules pass through a selectively permeable membrane in order to balance the concentration of other substances. In this picture you can also see that this is a semi-permeable membrane, so only some of the molecules are allowed to pass through the semi-permeable membrane. Alright, it is affected by concentration gradient and temperature both. When the concentration gradient will be higher, the rate of osmosis, the speed of osmosis will be higher. So it is we can say it is directly, the concentration gradient is directly proportional to the rate of osmosis at which the osmosis is carried out in the cells. Also, the rate of osmosis increases with the increase in temperature. So rate of osmosis is also directly proportional to the increase in the temperature. All right. So there is also a conflict. There is also a conflict that between few biologists. Few biologists take it as active transport, while some refer it to as a passive transport. So let's describe osmosis in a definition. The movement of water across a semi-permeable membrane in response to an imbalance of solute is called osmosis. Alright, so I have told you the relation also between the concentration gradient and the temperature, and the concentration gradient and the rate of osmosis also. Alright, so solutions that have a greater solute concentration will pull water via osmotic pressure. This depends on the total number of solutes and not on the type. Alright. Now talking about the tonicity. What is tonicity? Tonicity is a measurement of the effective osmotic pressure gradient. The water potential of two solutions separated by a semi-permeable cell membrane. So what is actually tonicity? The tonicity describes how an extracellular solution can change the volume of a cell by affecting the osmosis. Alright, so it is a measure of the effective osmotic pressure gradient. Alright, a solution's tonicity often directly correlates with the osmolarity of the solution. Now what is osmolarity? Osmolarity describes the total solute concentration of the solution. A solution with low osmolarity has a greater number of water molecules relative to the number of solute particles. Alright, therefore, a solution that is cloudy with cells may have a lower osmolarity than a solution that is clear. Alright, so there are three classifications of tonicity that one solution can have. First one is the hypertonic solutions, second one is the hypotonic solution, and third one is the isotonic solution. Let's discuss all of these. Now talking about the hypertonic solution. Alright, if a cell is placed in a hypertonic solution, there will be a net flow of water out of the cell. This is described in this picture as well. You can see the picture that these are the cells and the water has moved out of the cells. That's why these cells have shrinked. Now what is a hypertonic solution? A hypertonic solution can be referred to the solution in which the substance is put into the higher concentration molecule. So what happened? The water molecule present in the substance will move out and will flow from the lower concentration to the higher concentration. This is a hypertonic solution. So a solution will be hypertonic to a cell if its solute concentration is higher than that inside the cell. And solutes cannot cross through the membrane. Solute will not be able to cross through the membrane because of the big pore size and the big sizes, but the water molecule will flow out. Alright, so when we place any substance or any cell into a hypertonic solution, then the cells will shrink and its water molecule will move out of the cell. So this is hypertonic solution. Now talking about hypotonic solution. If a cell will be put into the hypotonic solution, there will be a net flow of water into the cell. Thus water will move inside the cell, and what will happen? The cells will swell. Take the example of raisins. Well, when we put raisins into the water, what happens? The raisins swell up because the water present in this compartment will flow into the raisins and the cell will gain volume. Now what is hypotonic solution? If the solute concentration outside the cell is lower than inside the cell, the solutes cannot cross the membrane, then that solution is hypotonic to the cell. You can see in the picture as well. Now talking about the isotonic cells. Now here in this picture you can also see that isotonic solution, if any cell will be put into the isotonic solution, they will remain as they were, because if a cell is placed in an isotonic solution, there will be no net flow of water into or out of the cell, and the cell's volume will remain stable. Iso meaning same, which is of same concentration. So when we put the same concentration together, then there will be no net flow because there will be no difference in the concentration gradient. Therefore, there will be no movement of any water. So what is isotonic solution? If the solute concentration outside the cell is same as inside the cell and the solute cannot cross through the membrane, then that solution is called as the isotonic solution. Moving on. Now talking about the mechanism performed by the cell called as homeostasis. As you can see, homeostasis is the condition in which the cell is able, or the living system is able to maintain its internal physical and chemical condition as it is. They won't get affected due to higher temperature or lower temperature or anything. The body temperature and fluid balance will remain as it is, as it was. Then this condition is called as homeostasis. Homeostasis is maintained by the cells of the body itself. This is the condition of optimal functioning for the organism, and the body is not affected by the body temperature and fluid balance, being kept with certain preset limits. Alright, so this is called as homeostatic range. For example, if in summers you see the temperature rises to 46 degrees Celsius, then also your body performs well, and in winters in the lower temperature conditions also your body performs well. So this is called as homeostasis, which is performed inside our body, that due to any physical or chemical condition our living system is maintained throughout. Now talking about transport mechanism. Alright, so plasma cell membrane is a barrier, but nutrients must get in products and wastage must get out. Alright, so this transport mechanism is also performed by the cell, in which keeping plasma membrane in mind, the nutrients must be absorbed by the body, whereas the products, the metabolic toxic products or the waste product must be removed out of the body. Now, how it is maintained? The permeability determines that what would be moving inside the cell and what is moving outside the cell, and let's nothing in or out is impermeable. Let's in anything passes freely permeable. Restricts movement is selectively permeable. All right. So plasma membrane is selectively permeable or semi-permeable. Which means it allows only certain molecules to move inside the cell and move outside the cell, and restricts other materials from crossing over. Either they will be needing any transport protein or ATP for moving inside or outside. Alright, so how the selective permeability is maintained? It is maintained on the basis of lipid solubility. That is, if any substance is lipophilic or hydrophilic, that is it is soluble into the lipids, fats or the water. And secondly, it will be dependent on the basis of the size or shape of the substances, that it is small or large, it will be able to move through the holes of the filtration membrane, and the electric charge it holds. All right, if it is polar, non-polar or charged. Now talking about carrier protein, because carrier protein plays a very important role in the active or passive in the transport, or if I will say in the transport of molecules. So carrier proteins are integral proteins that carry large non-polar, polar or ionic molecules across the plasma membrane. All right, down the molecules concentration gradient. All right, so this happens, the movement of molecules happen, the large molecules, the movement of large molecules inside the plasma membrane or outside it happens due to the presence of carrier protein. Carrier protein are able to transport amino acids, glucose and proteins that are very important for our body. If these important vital material will not be present, then our body will not function properly. So carrier proteins are helpful in transporting amino acids, glucose and proteins inside the cells for their absorption. They have specific binding sites. Due to the presence of specific binding sites, the protein changes shape to transport ions or molecules, and after the transport has happened, the transport has occurred, it is able to transport the amino acids, glucose and proteins. It comes in its original shape. Alright, so it exhibits the following characteristics similar to enzyme, that is, it is specific for a single type of molecule. Secondly, it creates a competition among molecules of similar shape, so as with which type of molecule it has to be attached. And the saturation, that is, the rate of transport is limited to number of available carrier proteins. If more number of carrier proteins are available, then the more number of molecules will be transported. If less number of carrier proteins will be available, then the lesser number of molecules will be transported. So saturation depends on the carrier protein. So these are some of the functions that are performed by the cell, and some of the mechanism that happens inside the cell is as well. So that's all about the physiology of the cell. Hope you are able to understand the cell structure of the cell and the physiology of the cell.